Hello, my name is Carol Leibel, and we want to examine the process of development and the effects that genes have on this process. We are going to emphasize multicellular organisms and really focus on the animal kingdom. So development is this wonderful process of time genetically controlled changes that occur in an organism's life, starting with a fertilized egg or a zygote. There are four main processes that take place. The first one is mitosis. And then once we have a mass of cells, we have to differentiate that cell. And also in development, we need to be able to establish different axes of the organism. And then we also have morphogenesis. So mitosis is going to result in an increased number of cells. Usually in the beginning, we're going to have a process called cleavage. And cleavage is a time of rapid mitosis without significant growth of the daughter cells. The cells become increasingly smaller as they divide, and each one of those cells is called a blastomere. And because this is happening so quickly, the G1 and G2 phases of the cell is shortened or practically eliminated. If you are looking at an animal cell, in general, mitosis occurs once every 24 hours. However, when you're talking about cleavage, usually cleavage can occur in seconds. So we have an example here in which here is a zygote, and that's a fertilized egg. And now we're starting to see division, in which here we have 4 cells, and here we have 8 cells. And you can see the volume remains the same, but every time the cleavage occurs, the cells are getting progressively smaller. Quite often at the end, we'll end up with a hollow ball of cells called a blastula. So what you're looking at down here is an actual embryo undergoing the process of cleavage. We have, or scientists have examined the development of nematodes. And the reason they've done that is that it's a very simple organism. It only has a total of 959 cells. So when you start off with one cell, that the fertilized egg or zygote, we can map how that one cell has turned into 959 cells and what they've become. Have they become muscles or gonads or nerve cells or what have you? And then what is also interesting is that during this process of development, 131 cells will end up dying. Now, some adult cells came into existence only after 8 rounds of mitosis, where other adult cells in the nematode requires 14 rounds of mitosis. An interesting process that happens during animal development quite often is called gastrulation, where we have this mass movement of cells. So you have this ball of cells, and now what we're going to see is that there's going to be this punching up, if you will, and you're going to have this mass movement of cells. That process is called gastrulation. And as you can see in this particular slide, is that gastrulation occurs differently for different kinds of animals. So in this example here, you're looking at a starfish, or a sea star. This is a bird, and then down here is gastrulation that occurs in a mouse. And so as I've just mentioned, the exact mechanism for gastrulation and how the cells end up can vary from animal species to animal species. And let me just mention something about plants. Plants do not have this mass movement of cells or gastrulation during development. And in part, that's due to the fact that they have cell walls, and so it's very hard for cells to move. However, in a plant, there are certain tissues that are devoted or set aside for cell division. Those tissues are called meristematic tissue or meristem. And in looking at a typical plant, we're going to find meristem in the tips of the root and the tips of the stem. That kind of meristem is called apical meristem because it's at the apex of the plant, and it causes the plant to grow in height or length, if you will. However, there's also meristem that's devoted to what we call lateral meristem and results in the plant increasing in the size of the girth. And the two types of meristem that have the ability to do that is vascular cambium and cork cambium. But as I mentioned, we're going to focus on animal development. Now, looking at a human being, and we are starting off as a ball of cells, but we end up becoming very specialized. And somehow we have to go from cells that look very much like one another to cells that are very differentiated. For example, we know that we have epithelial cells, and we have muscle cells, and we have nerve cells, connective tissue, and bone cells, and cartilage, and blood cells, and so on. So the question is, how do you start with this ball of undifferentiated cells to producing cells that are very differentiated? In humans, we have 210 different kinds of cells. The answer to that question is that these cells are producing different kinds of proteins. And as you all well know, genes are responsible for providing the information to make different kinds of proteins. So during the course of development, genes have to be turned on or activated, and other genes have to be inactivated. When looking at cells, there are two general classes of genes I like to think about. One is called those housekeeping genes that every cell has to have activated, that are necessary to go about the business of life. For example, genes that code for enzymes for cellular respiration are housekeeping genes because most cells have to have energy, and so these genes are activated. However, there are specialized genes that produce the unique gene product important for cells to differentiate. For example, it would be extremely wasteful if you had a liver cell and that liver cell was producing crystalline proteins that is needed for the lens of the eye. On the other hand, it would be very wasteful if you had a liver cell, if you had the lens cell, and that lens cell was producing an albumin product that is necessary for the liver. And so there has to be some sort of control element that will activate the genes at the correct time. Now, you know that there are transcription factors that can be used to activate certain genes or inactivate certain genes. So we want to examine that as we go through this process. As you can see with this little diagram, we have a liver cell, and here are transcription factors that are necessary to activate the albumin gene. And looking at a lens cell, here are a different set of transcription factors that are necessary to activate the albumin gene. And because these transcription factors are not found in the liver cell, then the crystalline gene is never transcribed. And the same thing is true up here, because these transcription factors are not found in the lens cell. The albumin gene will not be transcribed. In terms of development, not only do you have to have certain genes activated, but it also turns out that timing is extremely critical. Now, in terms of a cell becoming differentiated, we need to talk about a concept of determination. Determination are those things or processes necessary to commit a cell to a particular type of cell or a fate of a cell. Most often, when a cell is committed to a particular fate, it's usually irreversible. Once a cell is determined, then differentiation is going to occur. So let's look at this cute little diagram here in which we have a cell that is what we call pluripotent. Now, a cell that is pluripotent has the potential to make any kind of cell. And so down here, this represents the different possible outcomes. Now, as time goes on, different things are going to happen inside that cell. So at the end, that cell is committed to a particular fate, and once that happens, it cannot be reversed. Some terminology that you should be familiar with is a term called totipotent. A cell is totipotent if it can make any type of cell for the organism. And that includes extraembryonic cells. And extraembryonic cells or tissues include things like the membranes that surround the chorion or the amnion. Now, pluripotent cells are cells that can make any type of cell in the organism except the extraembryonic membranes. Okay, so if a cell is said to be pluripotent, it cannot make the membranes necessary for the chorion or the amnion. And then you have what we call cells that are multipotent. Now, a cell that is multipotent has the potential to make some different kinds of cells, but not necessarily any type of cells. And we're talking about usually stem cells. For example, adult stem cells and cord blood cells are considered to be multipotent because they can make different kinds of blood cells, but they cannot turn around and make nerve cells. Now, in this process of determination, what we have found out is that the older the cell is, the less likely that it is going to be pluri or totipotent. So in looking at this diagram here, we have a frog embryo and we have a tadpole. And over here on the left-hand side, they have taken the nucleus from a frog embryo and they have then put it into an egg that is what we call enucleated, which means they took out the nucleus. And now they're transferring it into this enucleated egg. And most of the time, that will result in a tadpole. Now, over here on the right-hand side, you have a frog tadpole. And what they have done is they have taken out a cell from the intestine, and they have removed the nucleus, and they have put it into an enucleated egg cell, and they have tried to turn around and produce a tadpole. Every now and then they will get a tadpole, but most are going to stop before the tadpole stage. You may have cleavage, but you're not going to have a fully differentiated tadpole. So in looking at this particular graph, they did this experiment over and over again using the nuclei from a late blastula, which means that ball of cells, and then they used the nuclei from an early gastrula, and that means after that large movement of cells takes place. And so they did it in this direction. This is an increase in time. And then looking at the question of whether you would get a tadpole or not, you can see that decreases as the cells become more and more differentiated. And so what that is suggesting is that as time goes by, more and more genes are becoming permanently inactivated that would be necessary to produce an adult tadpole, or to produce a tadpole, if you will. So as we mentioned, this concept of determination are events that lead to observable differentiation of a cell. And once determination has occurred, usually then the final fate of that cell is sealed. It's irreversible, if you will. And how we can determine, or how we know that a cell has undergone determination, is to take cells and to locate them into another location. So let me explain what I really mean from that. So let's suppose here we have an embryo and we have normal development, and we know that this area is going to result in the tail and that area is going to result in the head, and this is what the embryo is going to look like. Alright, so let's suppose that we have an early embryo, and we turn around and we take some cells that normally would result in a tail, and we move them or transplant them to another embryo to the head region. If they are not determined, then what will happen is that those cells will take on the identity of their neighbors, and they will indeed become head cells. Now, if the cells have been determined, it's later in development, and we remove those tail cells and we transplant them into another embryo in the head region, if it turns around and they continue to make tail cells, then what we know is that determination has occurred. So in this little diagram, we are looking at a blastocyst of a human, and these cells out here are going to give rise to those extra embryonic membranes. But these cells right here are called the inner mast cells, and those inner mast cells are said to be pluripotent at this point in time, and they have determined that they can give rise to the bone marrow, nerve cells, blood cells, heart, muscle, and pancreatic cells. And at this point in time, these cells are about 4 days old. When they get to be much older than that, then determination has occurred, and they no longer will be pluripotent. So now we know what determination is. The question is, how does it occur? So there are two sources responsible for determination. One is the use of what we call cytoplasmic determinants that are found within the zygote or the egg itself. And then another way a cell can become determined is by induction or signal secreted by neighboring cells. So here we have an unfertilized egg, and here's a sperm cell. And what you can see right away in that unfertilized egg is that there are two different kinds of cytoplasmic determinants, and they're not evenly distributed within the cell. fertilization takes place, and so again we have a zygote. And so now we're going to go through the process of cleavage or mitosis, and it produces a two-celled embryo. However, what do we notice? Is that the cytoplasmic determinants that came from the mother itself are not evenly distributed. And that has an effect on future development. Now let's look at this idea here. And once there is a multitude of cells, the neighboring cells themselves may produce signal molecules that can interact with a receptor site. And once that interaction takes place, can have an effect on the development of that cell. It can cause an activation of a signal transduction pathway and causing certain cells to be activated, not cells, but certain genes to be activated and other genes to be inactivated. So here is a little embryo. It's 32 cells, and these lower cells are producing some sort of signal that is going to influence the outcome or determination of the cells above. So let's talk about cell differentiation. What happens to cell differentiation after determination, or how that all works. We know that in order for a cell to become differentiated, there has to be a differentiation of genes. Some genes are going to be activated, other genes will be inactivated. That involves, of course, differential transcription, if you will. So there's going to be an appearance of certain messenger RNAs for certain specific proteins, and this may also involve transcription factors. And ultimately, once transcription and translation takes place of these activated genes, then we're going to see a change in the cell structure. This process of development, cell differentiation and determination, has been studied extensively in Drosophila, or the fruit fly. And here's the crazy thing. They have found out that many of these processes are homologous structures, that they also occur in humans. So you're talking about a fruit fly and you're talking about a human, and that we have some homologous processes taking place. That should indicate to you that development is an evolutionary conserved process. So let's look at an example of development in Drosophila in terms of determination and differentiation. So first off, here is the Drosophila life cycle. And we know that we have, this is a little male, this is a little female, and the male will deposit sperm inside of the female, and here's an egg, and it will become fertilized. And now what will happen is we'll have mitosis, 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 and the fertilized egg, by the way, will be laid. And what we will see is that there are 3 larval stages, and then this last, they are called, by the way, instars. This third instar, or third larval stage, will then we'll start to see a cocoon, or a pupa, if you will. And now what will eventually happen is metamorphosis will occur, and will end up, once it emerges, with adult flies. So let's examine pattern formation, or setting up the body plan once you have a fertilized egg. And because this is a bilateral organism, we need to establish where the tummy side will be, or the ventral side, where the back side of the organism is going to be, where the head end is going to be versus the posterior end, and then because it's bilateral, we need to know which side is going to be the left side and which side is going to be the right side. Those axes have to be established within the embryo. So the question is, how in the world does that happen? Remember when we talked about sources for differentiation, we talked about the fact that there can be what we call cytoplasmic determinants. And because they came from the egg itself, we call the genes responsible for those proteins maternal effect genes. Maternal effect genes are genes that, when you have a mutation, is going to result in a mutant phenotype in the offspring, regardless of the offspring's own genotype. So, in looking at the fruit fly, inside the egg, you're going to have messenger RNAs and ultimately proteins that are going to be produced from the maternal effect genes. And they are going to be made and produced in the egg while it's still in the mother's ovary. And as we said, a mutation can affect the outcome or the development of the little embryo. They have come to find out that maternal effect genes control the polarity of the egg and is very important in pattern formation. So when you have that egg, well, then somehow the egg is going to say, okay, this is going to be the back side, this is going to be the front side, the tummy side, if you will. This over here is going to be the head, the anterior end. This right here is going to be the tail. And then that's going to be the left, and that's going to be the right axis. Somehow, the egg itself is establishing that pattern formation. Now, we're going to focus on the anterior and the posterior axes. They have determined that there are two maternal effect genes called bicoid and nanos. So these two genes, bicoid and nanos, are used by, or not used, but they are activated, transcribed by the egg, and they are responsible for determining the anterior and posterior ends. Bicoid is responsible for the head end or anterior end, and nanos is responsible for making sure there's a posterior end. So it was hypothesized that in normal development, one might find a concentration of bicoid proteins at the end determined to become the anterior end of the larva, and it was also hypothesized that in moving from the anterior end to the posterior end, that the bicoid protein would decrease, and this would be just the opposite for the nanos proteins. And sure enough, that's exactly what they found out. The question is, where did these proteins come from? So you have an oocyte right here, an egg cell, if you will, and that oocyte is surrounded by what they call nurse cells that were derived from the mother, and they are responsible for secreting maternally produced bicoid and nanos messenger RNA into a maturing oocyte. Now, once those messenger RNAs are put into the egg, then we have to set up a concentration gradient. But the question is, how in the world does that occur? Well, it turns out that those messenger RNAs are differentially transported along microtubules of the oocyte using different motor proteins to transport the two different kinds of messenger RNAs. Alright, so let's look at this little example here, this little diagram. So here is the Nanos messenger RNA, and it has been, or it is connected to a motor protein that is different than the motor protein over here that's connected to the bicoid messenger RNA. Now this particular motor protein is going to transport the Nanos messenger RNA towards the future posterior end of the oocyte, if you will. And this particular motor protein is going to transport the bicoid messenger RNA to the anterior end. Once the messenger RNAs arrive at their respective ends, the messenger RNAs are going to become anchored into the cytoplasm, and now the messenger RNAs are going to be translated and produce bicoid protein and Nanos protein. And because the messenger RNAs are concentrated at opposite ends, what you're going to get is a protein gradient, as you see right here in this particular diagram. Alright, so here, over here at the anterior end, we have a concentration of bicoid messenger RNA, which is going to give rise to bicoid protein, and over here at the posterior end we have a concentration of Nanos messenger RNA that's going to give rise to Nanos proteins. Now here's the crazy part. In addition to bicoid and Nanos, they also have found two other maternal effect genes that are responsible for the posterior and the anterior end. And those are called hunchback, and hunchback is needed for the anterior structures, and caudal, which is needed for the posterior structures. But here's the crazy part. And before we leave, I want to show you this little electron micrograph of the larvae, if you will. This is a normal larvae. Over here is the anterior end that has the mouth parts and so forth, and over here is the tail end. If you have a mutation, of something like bicoid, what will happen is instead of having a head, you're going to have two tail structures. And that mutation is not happening within the larvae or the zygote itself, but that mutation occurred within the mother. And the other thing I want to tell you is that, explain to you a concept called morphogens. Substances that form a gradient in a zygote embryo and affects development are called morphogens. The bicoid protein and also the Nanos proteins are classified as morphogens. Alright, so let's get back to the business of hunchback and caudal. So here is the crazy part. We already know that if we're looking at the RNAs, that we know we have a concentration gradient of bicoid at the anterior end, and we have a concentration of Nanos at the posterior end. Okay? And we also said that we have hunchback and we have caudal. But when we look at those proteins, they form a gradient, but when we look at the RNA, look what happens. Those two proteins have an even distribution within the egg. So when we look at the proteins, though, we have concentration gradients. So how in the world could that happen if the messenger RNAs are evenly distributed? Well, that concentration gradient is due to the interactions of Nanos and caudal. And the way that it works is that the bicoid protein binds to and inhibits the translation of the caudal protein. So you're not going to find messenger RNA. So you're not going to find caudal protein at the anterior end. The nanose proteins binds to and inhibits the translation of the messenger RNA for hunchback. So now you're not going to find hunchback protein at the posterior end. So looking at this flow chart, if you will, here is the bicoid messenger RNA. The bicoid messenger RNA is found at the anterior end. It produces the bicoid protein. Now, the bicoid protein interacts with the caudal messenger RNA, and the caudal messenger RNA produces caudal protein. But you're not going to find it at the anterior end because the bicoid stops its translation. Now, at the posterior end, remember we have a concentration of nanose messenger RNA that produces nanose proteins. The nanose proteins interacts with the hunchback messenger RNA and will prevent the hunchback messenger RNA from being produced. So you're not going to find hunchback protein at the posterior end. So those two proteins are responsible for the gradient found with caudal and hunchback. So what we have just examined is we've examined the process of pattern formation in Drosophila, and now our next PowerPoint is going to look at morphogenesis in as we see it in looking at the fruit fly. So we'll talk to you later, and please view Development Part 2. Bye!